talking about something that may make sense or may not make sense. How many people tried the quiz? All right. How many people feel like you've taken on the quiz and you got it? All right. How many people feel like the quiz has got you? Uh-huh. And so in 50 or 70 easy steps, right? It seems like that this quiz goes on forever. When does it due? Oh, good. You got time to deal with it. Now, the only bad thing about putting a quiz, far away in the future means you have to think about it for a long time. So, the further it out, the more you have to think about it. But what happens and what I want you to see is that the book teaches all of the reactions in perfect stoichiometry. Okay, but the body very rarely, you very rarely eat in the perfect stoichiometry. Okay, so what happens is the body will do as many reactions as it can, and then it may stop because it runs out of one reactant. That's what you're going to end up at the end. You will have a couple of intermediates that will be one of the intermediates of the pentose phosphate pathway, and they won't have anything else to react with. Okay? Now, at times it'll look like it will be never ending, but you will get to a point where you get to a couple of things that just don't react with each other. That's when it stops. And the body will wait until you eat more and go down that pathway, and they will just be there ready to continue the reactions. And what I wanted you to see was that you didn't need 3 whole ribose equivalents to go through the non-oxidative, because 2 reacting can go a few steps, create a fructose, which then could go to glucose, which then can go all the way around and enter back in. And so it will use these pathways interchangeably and move. As long as you have reactants, it will continue to do reactions. It doesn't have to go to full completion, but it can react as far as it can until you have something you can't react anymore. All right, most of the reasons why we might integrate the oxidative pentose phosphate pathway, the non-oxidative, which is transketolase, transaldolase, and glycolysis or gluconeogenesis, using those 3 pathways together will be dependent upon what the body lacks. Okay, we're trying to keep homeostasis, and so if I need one thing, I'll run one direction to make that one thing. Or if I need another thing, I'll run a different way. Or if I don't need things, I can't go through some pathways, but I can still go through other pathways. This is how the body reacts, and it is part of regulation. A lot of times regulation is just concentrations trying to keep a certain level, meaning that things will only bond or bind into enzymes effectively if it's at a certain level. If it doesn't bind effectively, it's not going to react. If you have too much of one thing, then it binds effectively to an enzyme and it will progress down one pathway or another, so it lowers that particular concentration to make it that concentration that you want. Most of the regulation that is based upon concentrations is an allosteric regulation, where these concentrations will affect the enzymes, either making them go or stopping them, based upon the availability of the substrates, and or if a regulating molecule from another pathway will tell one enzyme to either go or stop. An example would be regulation of the pentose phosphate pathway itself. One of the main allosteric regulators is either the absence or the presence of NAD plus or NADP plus or NADPH. Okay? And so glucose can go down the pentose phosphate pathway, or it can go down glycolysis when we are degrading glucose, and which direction it goes will be dependent upon how much NADH is there or not. And so I will go down the glucose the pentose phosphorate pathway if I have a lot of the NADPH plus. What I do want to have is a really large source of NADPH available so that I can do synthesis. If I don't have that, then glucose gets used first to make sure that concentration goes up. So the enzyme that starts the pathway for the pentose phosphate pathway, the oxidative portion, okay, is regulated by the fact that it has a lot of substrate, and it'll make it go down the pentose phosphate pathway. If by chance I have a whole bunch of this floating around, then it will bind to this enzyme and slow it down, meaning that the glucose becomes available for glycolysis. And so the big regulator allosterically is either the absence of NADP plus or the presence of it, and the absence or presence of NADPH. All right. Now, why in the world do we have the pentose phosphate pathway, and why are we actually creating NADPH? The first and foremost reason is for synthesis, but that is not exclusive. The second most often used reason that we use NADPH is to fight oxidative damage. Okay? Oxidative damage means I have a very reactive oxygen species. And so a lot of times we get radicals formed. Okay? A lot of times these radicals are misreactions or things that escape the oxidative phosphorylation, the electron transport chain. Some of those electrons get loose, react with oxygen, and then that oxygen can react with lots of things because it's so reactive. It can react with our DNA, it can react with proteins directly, it can insert itself into something that is really not reactive, like a carbon and a hydrogen bond, which usually doesn't react with very much, but a radical can. And so if it gets into your DNA and reacts and messes things up, that's not good. It could change or mutate your cell. And so we try to have mechanisms in the body to minimize how many radicals you have roaming around, or the reactive oxygen species. Now, we know that we replicate cells, right? We replace cells, and there's a statistic that says every 7 years we replace every cell in your entire body. Well, if you replace every cell in your entire body with every 7 years, why in the world do you age? You have brand new cells being created all the time. One of the theories of why we have aging is that there's an accumulation of errors from radicals. And so those get propagated, and so what is copied is not a perfect copy, it is an error-prone copy. And so to protect us from aging, we put in antioxidants. That's why in every face cream that you have, there are antioxidants, because it is going to keep you younger, okay? And so, and if they put a scientific name in their product line, then it's more expensive, okay? And so the NADPH can be used, okay, to create that NADPH, and then it will be given to glutathione, okay, and we studied that particular molecule before, and the glutathione is a carrier of hydrides, and that glutathione will take the hydride and react it with the peroxides that are formed from the radicals to create a detoxified water, which is necessary and not reactive enough to cause damage. And so a lot of our NADPH is used to neutralize reactive species. Now, oxidative stress is something that happens all the time, but oxidative stress can be brought upon by taking drugs or eating certain foods. And so sometimes if you take anti-malarial drugs and or eat certain foods, you increase oxidative stress. Okay? And the oxidative stress in normal, in people that have normal functioning pentose phosphate pathway, we have enough NADPH to take care of that extra reactive species. But if by chance you have a deficiency in the pentose phosphate pathway or the first enzyme, you don't make as much NADPH. Okay? Now that deficiency could be total absence, but most likely just not one that runs as well. And so if by chance you have a deficiency and you undergo these stresses, these extra stresses, then you cause an illness that you may not have known before. That illness could be also brought upon by smoking, and that causes oxidative stress also. And so there are some people that cannot take antimalarial drugs because of a deficiency that they did not know existed before they started that. All right, that's the end of the pentose phosphate pathway. Remember, we're taking glucose and we're manipulating it. We're either breaking it down in a different way than we saw before, the glycolysis, or we're going to synthesize it from things that we didn't see before, and that's what we're going to talk about, gluconeogenesis, and then we're going to store it. That's the last thing that we're going to talk about in this chapter. Okay, we have talked at gluconeogenesis, but we haven't taught gluconeogenesis, so we're going to do it now. It is essentially an idea, it's the reverse of glycolysis, but it is not absolutely the reverse. It is 70% the reverse, okay? Of 10 reactions in glycolysis, 7 of them are in equilibrium, okay? So that means they can go either way. That means if I give it lots of product, it will go towards a reactant. If I give lots of reactant, it'll go towards product. So 70% of the glycolysis is gluconeogenesis. So you know 70% of it. So you got to see right now without even learning anything else. Okay? That is great. There are 3 arrows in glycolysis that are one direction. Okay? That's not a musical group. Now, when you take a look at those, that is every time that we actually use or produce ATP. Okay? Those are such large changes in energy when I break ATP, I just can't reverse that. Okay? So hexokinase, one direction. Fructose 6-phosphate to fructose 1,6-bisphosphate. Again, a single arrow in one direction. And then when we make the phosphoenol pyruvate into pyruvate and we make ATP, again, that is a very large energy, one direction in glycolysis. So these have to be bypassed in gluconeogenesis, and we bypass the 1, 2, 3 reactions with 1, a different enzyme, 2 reactions with a different enzyme, and then 3 and 4. This is such a large change in energy, I actually have to use 2 different reactions to go from pyruvate back up to phosphoenol pyruvate. Okay? So I have 4 separate reactions in gluconeogenesis that are separate from glycolysis. 2 of them are simple, 2 of them are a little bit more complex. Okay? Gluconeogenesis, it really is essential because the red blood cells in the brain use glucose almost exclusively, and the brain requires about 120 grams of glucose a day by itself, and all the storage molecules in your body at any one time only has 190 grams of glucose. So if you do that calculation, you would die in 2 days if you didn't eat anything. Now that's not true, is it? You can go on a fast for longer than 2 days. So that means we have to have a mechanism to synthesize glucose in our bodies without taking anything else in. This is where gluconeogenesis allows us to fast longer than 2 days. Now, let me tell you, that doesn't help people from getting hangry, okay? But it will help you live. Just no one might not like to be around you during that time, but all right. Now, where gluconeogenesis actually occurs is really in two places: the liver and the corteny of the cortex of the kidney. These are the two. These are the two. Thank you. These are the two tissue types that actually will cause most of the gluconeogenesis. Now, where in the world do we start gluconeogenesis from items within the body? All right. Lactate is the first place because lactate was the end product of anaerobic oxidation of glucose. If you run out of oxygen, the best you can do is take glucose and change it to lactate. Lactate is built up in the muscle. Muscle can't do anything else with it except make you cramp up and make the burn feeling, okay? And so if I leach that out of the muscle and get it back to the liver, I can make glucose again. Second, amino acids. This is during starvation, most of your glucose that is for your brain is converted from protein, okay? Now, we don't store protein in any significant amount. Proteins have to be synthesized from the transcription translation process, and so we make protein, but you have a large fraction of muscle in your body of which you can sacrifice that muscle, still move around, but actually convert a lot of those carbons into glucose itself. The last thing is glycerol from fat. Now, a traditional fat has 3 fats on them. Most fats are 16 carbons long, plus a 3-carbon alcohol that is called glycerol. That is a triacylglycerol. That is 51 carbons. 51 carbons is a whole lot of carbons. Only need 6 for glucose, but of the 51 carbons in fat, only the 3 from glycerol can be converted into sugar itself, into glucose. The other 48 cannot. And so this is a limitation in terms of how we can manipulate our stores of energy. Our largest store of energy is within the fat cells, but the majority of that energy cannot be converted into glucose. All right, so these are where we're going to start the synthesis of glucose from these 3 sources. All right, the 3 irreversible steps are important because when I go from glucose to pyruvate, if you do all the delta H's, it gives off about 20 kcals per mole of energy, okay, a lot of energy. The gluconeogenesis has all of the enzymes except the 4 enzymes that bypass the 3 irreversible steps. Here are the 2 simple ones. First, glucose 6-phosphatase. The enzyme phosphatase does what? Breaks off a phosphate. I go from glucose 6-phosphate to glucose. Okay? One thing about this, glucose comes into a cell and gets phosphorylated immediately, so it's trapped inside the cell. Once you take the phosphate out, glucose has the ability to go outside the cell back into the bloodstream. Okay? On rare occasions that occurs, and really the only tissue type that allows that to occur is liver. Okay? Here's the next one. Fructose 1,6-bisphosphatase. Again, I'm going to take off a phosphate. I can convert the 1,6 into the 6-phosphate, heading towards creation of glucose. So 2 phosphatases are going to be the opposite to the 2 kinases in glycolysis. Okay, here's where it gets complex. pyruvate must first be converted into oxaloacetate by, really, if you take a look at the molecule, okay, here is an acid, there's the acid. Here's a ketone, there's a ketone. Here is a methyl, there's a methylene. Okay, and there's a CO2. By the enzyme pyruvate carboxylase, I put a carboxy on a pyruvate, I get to oxaloacetate. We have seen this reaction before. Okay, this is a reaction in which I can take some of the pyruvate and makes oxaloacetate so that I can refund some of the molecules of the citric acid cycle and allow another pyruvate to go to the acetyl-CoA to enter in as citrate. So we've seen this reaction before. But if I'm not going to use this for the citric acid cycle, then I can use the phosphoenol pyruvate carboxykinase. Oh my goodness gracious, what in the world is that? This CO2 is missing. Here's the methylene that has 2 hydrogens. And I have a double bond, which is right here. And I have a phosphate on an oxygen. What happens is I will form CO2, I will form the double bond, and that attacks GTP at the gamma phosphate so that GDP is a leaving group and CO2 is a leaving group. And I get to the structure of phosphoenol pyruvate. From that point on, I have equilibriums, and I can go up through gluconeogenesis. We've seen this particular reaction before. There is a series of enzymes that work, and so I activate a CO2 that's going to be added. I then swing it over to the active site where pyruvate is because it's got this long chain that can move it around to create the oxaloacetate. And so this enzymatic reaction we've seen before. Okay. And the pyruvate carboxylase itself occurs inside the mitochondria. This oxaloacetate has the ability to go to the citric acid cycle if I need it. But if I am regulating for gluconeogenesis, it is going to go to malate, which is the opposite conversion in the citric acid cycle. And I do malate because that can be transferred out. The malate can be transported. And then the malate has to be converted back into oxaloacetate. When I have oxaloacetate in the cytosol, that indicates that it's part of gluconeogenesis. Okay? And then we can start up the process from there. Okay? Now, a lot of times, the anaerobic exercise that allows glucose to go to lactate, okay, as the end product, I make just a very tiny amount of ATP. Lactate stays in the muscle until it leaches out, gets into the bloodstream, will float into the bloodstream, and then be accepted by the liver. The liver will then has an enzyme called lactate dehydrogenase, and it will convert it back into pyruvate, which then allows it to go through those processes through gluconeogenesis to get back to glucose. I actually have to invest quite a bit of energy. I have to invest an NADH, 4 ATPs, and 2 GTPs to get this lactate back to glucose. So I've only extracted a few, 2 ATPs here. I got to lactate, then I come back over here. I have to invest all of this energy to get back to glucose. And then I can extract all the energy from glucose, but the body has already used quite a bit of the energy. It is a waste product, but it is something I net some of the energy back rather than losing all of the energy itself. The process by which lactate goes through the blood to the liver, going back to glucose, which then can go back to the muscle, is called the Cori cycle. Dr. Cori described it. The enzymatic reaction for the oxaloacetate to the phosphoenol pyruvate, I already showed you, form the CO2, form the double bond, attack the GTP at the gamma phosphate. And we've got the phosphoenol pyruvate. All right. Regulation. In this case, we have both allosteric regulators and hormonal regulators. Okay? In groups of 3 or 4, I want you to look at this slide here, this slide here particularly, and this one. And I want you to identify why each of these regulators either slow down or speed up glycolysis or gluconeogenesis. Why would that be the reason? Okay? In your group, be prepared to explain why some of these, and I might ask you to which one, why that particular regulator would either slow down gluconeogenesis, speed it up, or slow down glycolysis or speed it up. Okay? So break up into small groups, and I want you to identify all the different regulators and why this might happen. Okay. All right, so let's look at each one of these things. All right. What in the world is causing things to go or not go? All right. And so we have lots of regulation between those two enzymes. That's the glycolysis and that's the gluconeogenesis, right? And we have lots of little molecules that are affecting these, gluconeogenesis, and this is glycolysis. Okay? And so let's think about this. All right. A big regulator for gluconeogenesis or for glycolysis is this fructose 1,6 bisphosphate. Makes it go. All right. Is this molecule a natural product? I mean, is it part of glycolysis? Is it part of citric acid cycle? Is it part of gluconeogenesis? The 2,6. That's not one of the structures. Well, why was 2,6 actually made? Okay. Well, where in the world is 2,6 actually made? 2,6 is made by this protein, and it changes fructose 6-phosphate into fructose 2,6 bisphosphate. Well, what made this go? High blood sugar, insulin. So if there is insulin, that means I have lots of sugar. It made this enzyme function and made this molecule. There's lots of sugar. Do I need to make sugar? There's lots of sugar. There's a hormone response that says, make the breakdown of sugar go. So that glucose 6-phosphate, this fructose 2,6, is a product of the hormonal regulation that says, I'm well fed, I have lots of starting material. Makes this enzyme function, makes a lot of this, and makes glycolysis go. Okay, got that? And so that is a hormonal response. Well, over on this side, all right, if you have a lot of this, it stops it. Well, if I have a lot of sugar, it's going to stop the gluconeogenesis because the hormone response says, I already have lots of glucose, don't waste all that energy making it. So that seems pretty cool. Here, to go down glycolysis, not only does it upregulate this one, which forces it to go down, it also regulates this one, which makes that last step of glycolysis. So insulin causes those two to be made and glycolysis to go. So that's great. All right. If I look at AMP, okay, there's AMP there and there's AMP there. All right. What does AMP tell you about the cell condition? What is AMP? It's the mono. Does that mean I have lots of energy or a little bit of energy? Okay, a little bit. Because ATP says I have energy, right? AMP says I don't have energy. If I don't have energy, the way I can make energy is making glycolysis go, right? If I break down glucose, eventually I go through citric acid cycle and I go through oxidative phosphorylation and I make ATP. So AMP will make it go. That seems logical. What about AMP over here? I don't have a lot of energy. Why does that stop gluconeogenesis if I don't have a lot of energy? It takes a lot of energy to get it this direction. If I don't have ATP, I'm not going to make glucose. Okay? That just doesn't seem like it's going to be a good thing for me, right? All right. So I've got that. Ooh, ATP. That means I have lots and lots of energy. It slows down glycolysis. Why? If I already have lots of energy, do I need to break down glucose? No, not really. Okay. Here, ATP. If I have lots of energy, do I need to go down? No. ADP is the same as AMP. That represents not a lot of energy. Not a lot of energy, I'm not going to go up. Okay. Okay, citrate. Ooh, what is that? Where do I find citrate? Yeah, the citric acid cycle. And what feeds citric acid cycle? Glycolysis. Okay. So, what if I have very little citric acid? Then what does it say? If I don't have a lot of citric acid, maybe I take my glucose and make it go. All right? But what happens if you're building up lots of citrate? Well, that means you're feeding stuff in and I get citrate. It means it's not going around, right? If I get big citrate, that means this is totally stopped. Well, if I get lots of citrate, what is it going to do? It says, hey, don't break down. Let's make some. Okay? Do you see how we're doing that and justifying all of those? You got a couple more things to take a look at, but those are the kinds of things you need to think through. Okay? It is powerful, yes. Very, very powerful. Yes. It's the same as you learned before. It is the same thing. Yes. Correct. but we learned it only for glycolysis last time. Now we're adding in, it will also control gluconeogenesis. Yes. Okay, so the significance of alanine. There are ways that you can extract energy from glucose, from your sugar stores, or you can extract energy from fat and from protein. If by chance you're taking your muscle, breaking it down, putting it into the bloodstream, and it's coming in, all right, then you're starting to use your muscle stores because you need to conserve your sugar for the brain. So if you have protein floating around, then you might not want to break down glycolysis anymore. You want to use the sources of protein and fats at a certain moment in time. Fats are represented by acetyl-CoA, or all fatty acids, the big chain, and proteins are monitored by, could have been any amino acid, but alanine is one that the protein, the other enzymes are sensitive to. All right. Are we ready to explain it to me? No? No? It depends upon which one you ask. Okay, so which one do you want to explain that is easy?
I guess
The acetyl-CoA. Okay, so why would the acetyl-CoA, this is the one that you chose, why would it tend to make the gluconeogenesis go up?
You need it to be able to get to the oxaloacetate.
Okay, you need it to be able to get to the oxaloacetate. If you have lots of acetyl-CoA, then I could conceivably need oxaloacetate to react it to go into the citric acid cycle. So a lot of the reactant could use this, but it also could indicate that I'm breaking down fat and using fat for energy in the citric acid cycle. And that means if I'm using fat rather than sugar, well, that means maybe I might need to make sugar for my brain. Okay, if you've transitioned over to fat stores, then you're conserving sugar, and if you're conserving sugar, you may need to synthesize sugar for your brain. Okay, that seems good. Vabi, what does your group want to explain? Okay, let's talk about fructose 2,6-bisphosphate. Now, is that a hormonal signal or an allosteric signal? Allosteric? Hormonal? Okay, it's kind of in between. How or why is fructose 2,6-bisphosphate made? Why? What would the signal in the body 2,6-bisphosphate? Why in the world would we make it? Other slide. No. Not that one. Yep. Blood sugar. Blood sugar is what? High or low?
I mean, depending on which one it is.
Well, we're making the 2,6. So what causes 2,6 to be made? High sugar. And so insulin is a signal. That's a hormonal signal. And the hormonal signal creates the version of the protein that makes 2,6. Okay? And so usually an allosteric is a normal protein in one of the pathways gets kind of big or gets small. 2,6 is kind of oddball because it gets made because of a hormonal signal. Okay, so if 2,6 is there, that meant insulin was the hormone. That meant I had a lot of sugar in the blood. So what would it cause glycolysis to do? The 2,6. Break down, right? Because I have a lot of sugar, let's break it down and make as much energy as possible. Okay? On the opposite end, okay, so we've explained this one. On the opposite end, if you don't have 2,6, what was the hormonal response that caused that to happen? Glucagon. Now, this only happens in the liver because glucagon is a starvation signal, and the liver has a receptor for glucagon, muscle does not. Okay? And so, if I don't have 2,6, that means I didn't have sugar in the blood. If I don't have sugar in the blood, my brain is going to not like it. And so what does that force the liver to do? It makes it what? It slows down the, let's see, fructose 2,6-bisphosphate is going to, oh, okay. Oh, if you slow it down, you're not going to have this. But if this is present, that is in the signal of insulin. If I have insulin around, I have lots of sugar, don't go up gluconeogenesis. Okay, so if this is not there, I'm going to go the opposite direction. Okay, so we've got that. All right. Did you do a 2 group or did you do a 4 group? 2 group. Which one do you want to explain?
I mean, we'll take ATP.
Okay, ATP. All right, I've got ATP right here. All right, do I have ATP anywhere else? ATP. Why does ATP make this slow down, the glycolysis slow down?
Because glycolysis is just taking glucose, making ATP.
Right. So if you have lots of ATP, don't need to do it. There you go. All right. You are group of 2? Which one do you want to do? ADP. Great. Okay, let's see where ADP is. You've got ADP here. You've got it there. And that's it. ADP in 2 places. Why does that slow down gluconeogenesis? Yes, which is what? What did they say? It is the opposite of what they said. If you don't have energy, ADP is a measure of the cell not having a lot of energy, right? And so we want to make energy, right? Also, gluconeogenesis requires a lot of ATP. And so if you don't have a lot of ATP, then you probably are not going to make the glucose. And so ADP tends to make the gluconeogenesis go down. Okay? All right. In the front, what do you want to explain? There was another ATP. It was the same as what they said.
We'll do AMP.
You will do AMP. Okay, AMP represents what in the cell? Yeah, well, if you have lots of AMP roaming around in the cell, what does that make your cell? Does it have lots of energy, or does it have little energy, or does it have very little energy? It has very little energy. Okay, so right here, very little energy will stop making glucose. But very little energy, the molecule that says very little energy, makes what go? Glycolysis, because I want to make energy. All right. Do you have a group of 4 right here? All right, you're running out of stuff.
I know.
So which one do you want to do? You got alanine, you got citrate. What's your feeling? You feel lucky?
Let's go for citrate.
Okay, let's go for citrate. Okay.
So you need citrate to make oxaloacetate?
Do you need citrate to make oxaloacetate?
I mean, it helps.
You need oxaloacetate to make citrate. Acetyl-CoA and oxaloacetate make citrate.
Glycolysis, you can get there.
Well, if you go all the way around, you can get there. Yes, that is true. Okay, we're talking about glycolysis and gluconeogenesis, right? Where is citrate made? Citric acid cycle, right? What I'm feeding. Okay, so glycolysis goes to citric acid cycle. All right, the first product of citric acid cycle is citrate, right? Okay, what happens if citrate starts to build up in concentration? Well, that meant the citric acid cycle is not moving around. But that meant you are still running and feeding pyruvate into the citric acid cycle, and you build up citric acid, the citric acid itself, but it can't go any further. If you get lots of citric acid there, what happens to the glycolysis and gluconeogenesis if you have a lot? It tends to say, don't break down glucose and feed it. It says, hey, I have a whole bunch of energy because I'm not making any more NADPH or NADHs. So if I have a lot of citrate, that means I really have a lot of energy molecules. Let's make some glucose. Okay? So citrate upregulates the gluconeogenesis and it downregulates glycolysis. Okay? You don't have much choice. Why in the world does alanine stop glycolysis? Yes, but why? That's what it does.
So if you have low amounts of alanine, it's kind of almost like...
Well, no, you have high amounts of alanine. It's going to slow this down. So this is saying I have alanine, slow this down. Okay.
Isn't it just kind of a signal that you have like a lot of other precursors as well?
Yes, it is. Remember, we can get energy from 3 sources, right? We can get energy from sugar. We can get energy from protein. We can get energy from fat. If by chance in your muscle you start having protein being broken down and you got single amino acids, they're going to be broken down. It's now going to start using the protein and conserving the glucose. All right? If you had the presence of fat, it will also slow down glycolysis because I have another source of energy. And so glucose is the first quick source, and you start using, or when you exercise, you get your second wind, you start using fat and protein to the exclusion of glucose. All right? Depending upon how well conditioned you are, it's how quickly your metabolism shifts to using those other two and conserving glucose. And so a very fit athlete will be able to transition, and part of their conditioning is being able to mobilize and move their metabolism reactions from one source to the other much more quickly than the couch potato. Okay? And that's part of the conditioning. All right, now remember that the fructose 2,6-bisphosphate, which is produced from a hormonal signal, is a hugely powerful regulator, and it can upregulate things and downregulate things in terms of not just twice, but 15 or 25 times. Okay, it is a very, very big influencer. Hormonal responses are usually large changes in activity. Allosteric responses are usually small kind of adjustments. And so fructose 2,6-bisphosphate is directly from the hormones creating large changes. And then all these other ones that we talked about kind of tick it up a little bit or slow it down a little bit. It doesn't go from 0 to 100%. It doesn't make that kind of change. All right. Now, insulin can affect the production of the 2,6 in response to your hormones. The insulin will affect both liver and muscle, and it tends to make glycolysis go. But glucagon only affects the liver molecules, and it will tend to make gluconeogenesis more active. All right? Which would be exactly the opposite of what you would think muscle, but liver does function exactly opposite. It doesn't use glucose for its own energy. It actually feeds glucose to the bloodstream to make sure your brain keeps functioning. Okay? All right. There are some other regulators for the phosphofructokinase. Protons is the additional one. It prevents the continued formation of lactate. And so if by chance you're producing a lot of protons, that means you're doing anaerobic oxidation. You're making lactic acid. And that tends to be a very wasteful reaction, and so it tends to slow down glycolysis because it's an indicator that you're not doing it with oxygen. We've already talked about citrate. We've already talked about ATP. Glucose-6-phosphate, the phosphatase, that last thing, is controlled in a totally different manner than what you've seen up to this point in time. Okay? Its control is not necessarily a covalent modification. It's not an allosteric issue. It is all about compartmentalization. We control its activity by controlling, does it have substrate or does it not have substrate? And so the inside liver, okay, the enzyme is found only inside the lumen of the ER, okay? Because when we get glucose inside, the hexokinase, we want to phosphorylate it and keep it and use it most of the time. But if by chance we've got gluconeogenesis running, we give the glucose and transport it inside the ER, creating glucose itself, and then we send out a whole bunch of it, which then can be transported out. Now, there's a competition. When I send this out, some of it can be rephosphorylated. That's not necessarily good for the overall process, but we overwhelm and we feed this out to glycolysis. So access to this molecule, or the use or control of the transporter, is what controls this, not a regulatory event that we normally talk about. So it's access or compartmentalization, or if it's sequestered. So that's kind of an odd thing. Okay? Overall, gluconeogenesis, negative 9 kcals per mole. It is not the absolute reverse of those three steps. If I actually reversed it, it would be 20 kcals per mole in the positive, meaning that it would not be spontaneous. All right. So we have made glucose from lactate. We have made glucose and controlled the gluconeogenesis and compared that to glycolysis. All right. The last thing that we need to talk about within the mammalian cell system is how we store glucose. We store it in the form of glycogen. There are two main places glycogen is found. The liver has a much higher concentration of glycogen per gram. It is like a big old sponge of glycogen. There's a much lower concentration in your muscle, but because you have so much more muscle, that's where most of the glycogen lies. Glycogen in muscle, or the muscle itself, if it actually traps fat or if it traps sugar, it is a selfish tissue. It will never let go of those energy molecules. It may store it for a little time, but it is going to break it down. Liver, on the other hand, when it traps glucose, it holds on to it and holds a very large store of it, but it's intended to let it go and be released back into the bloodstream. And so, glycogen has a higher concentration, but the largest amount is in muscle. Glycogen is a polymer that is branched. We've talked a little bit about this before. It's an alpha-1,4 glycidic linkage with a branch point on average every 10. It is not every 10, it's on average every 10. Glycogen, when broken down, is kind of interesting. It is going to produce glucose 1-phosphate. Now, take a look at glycogen. What do you not see at all in here? Do you see any phosphates? Not at all. When it breaks down, it is going to break down into a glucose that has a phosphate on it, but I'm not going to use ATP. Okay? When we first looked at glucose coming into the cell, the first thing it reacted was hexokinase and used an ATP. That used energy. Glycogen, when it breaks down, is going to break down in an enzyme that will be able to put a phosphate on without using energetic molecules. Now, glucose 1-phosphate is not part of any pathway that we know of at this moment in time, and so we're going to have a mutase where it moves from one to another. Okay, it's going to go from the 1 position to the 6 position. Now, we've seen a mutase mechanism before, right? We moved it from the 2 to the 3 position. Did the phosphate from the 2 move to the 3? No. What was the intermediate? You had both of them phosphorylated, and then the phosphate that gets moved is a totally different one. Same kind of mechanism. That's how mutases work. Okay? Once it's glucose 6-phosphate, we know now it can go down pentose phosphate pathway if I need that. It can go and create glucose to go to the bloodstream if you're in liver. If you are in muscle or the brain, it gets broken down to pyruvate, and depending upon what's going on, if you have oxygen or not, I can get all of the ATP if I go to CO2 and water, and I get part of the ATP if I go down to lactate. This is the aerobic, and this is the anaerobic pathway. Okay? Now, glycogen is stored in very large particles, large enough that I can actually see with a light microscope. Most of the things that we talk about in the biological system, when we talk about enzymes, they're so small that you can't see them with a light microscope. But this thing is huge, okay? Because it's a collection of things. It is the glycogen particle itself, but it has every enzyme necessary to break down the glycogen and every enzyme that is needed to make glycogen bound all together. Okay? This creates the ability to be very quick in processing glycogen, but this also creates something that we have to really fight against. We have to make sure that we never have the synthesis and the degradation going on at the same time. Okay? Since they're in the same compartment, bound to the same substrate, we're going to talk about something called reciprocal regulation. One signal will turn one pathway on, but at the same time turn the other pathway off. Okay? Because we have to keep tight control. If not, we would have a wasteful product. Okay? And so this is really common, and it allows us to have very quick responses. Okay. The enzyme that will do the degradation, the majority of the degradation, okay, is the enzyme glycogen phosphorylase, but a lot of people will just call it phosphorylase. Okay? The reactants are glycogen plus an inorganic phosphate. All right? And what happens is the terminus, the last sugar of an end, will react with the PI and create glucose 1-phosphate. It will break the alpha-1,4 bond. Okay? Then the mutase will change the glucose 1-phosphate into glucose 6-phosphate. Okay? Now, it's important. I make a phosphate bond without the consumption of ATP. Glucose is given off in the anionic form, a phosphorylated form. That means it stays within the cell, so that's good, and it can be converted into a useful product. Now, what happened here, if I monitor the reaction, okay, So I have a little reaction, okay? This is time, and this is the product, okay? What will happen is I will have a very fast production of product over a very short period of time. Then the enzymatic changes, and it slows down, okay? Can be very, very quick. It's called a burst phase. The phosphorylase will be able to break down every end of the glycogen at the same time. And it'll break off lots of glucose 1-phosphates, glucose 1-phosphate, glucose 1-phosphate, until it gets within 4 residues of the alpha-1,6 branch point. Then it stops. Why? Why would it stop? It was happily chewing away at the end of these sugars, and all of a sudden, the phosphorylase doesn't break it down anymore. Do you have any clue why that might happen? Take a look at the original substrate. What is the structure? It's a straight what? Straight line, right? But when you chew it all down, you get to something like this. What's wrong? Okay, why would an enzyme not produce any more product? Change shape, what was it? The substrate doesn't fit. Remember, for an enzyme to work, you have to have the substrate bind to the enzyme, right? It has to fit. It has to be the right shape. If it's long and straight, it falls down and fits into the active site, and I can break it down. But when I get to this, these two chains are too close to each other, and it can't fit into a groove that can only hold a single straight chain. The enzyme is, for conceivably that point, dead. It can't bind to the substrate anymore. But I had lots and lots of ends. I produced lots of glucose in a very quick fashion. All right? Before I can go again, I have to use another enzyme called the debranching enzyme, or the transfer, and I'm going to use the transferase activity of that enzyme. And it will take a block of 3 and move them to the end of the other chain. I now have a single alpha-1,6 sugar. That branching enzyme will then cleave that alpha-1,6, but it comes off as glucose. What is different about that from every other glucose coming off? No phosphate. Okay? And so I give off glucose with no phosphate.